Just hang out on air just live, and regardless, you should record. So
okay,
let's hope that it's recorded. So
and it's listening to you.
It is. Let's hope this is working. Yeah. Allegedly. Alex, you put a walking around. Okay, no, I'm just gonna try and center it.
Okay.
Welcome. We are here to hear from Alex Lowe, as you know, and we have an hour or so to listen to what he has to say, but let me tell you, he has had a lot to say in the past. I am just going to scroll through these scientific efforts, mostly out during his graduate school career. 4 of them first authors, 10 altogether. Many co-authors sitting in the room. And he also writes at the interface of science and the public. He was 2016's most prolific poster on SciPause. Started Sound Scholars and won second place in 2016 in the 1,000 word challenge, which requires that you can talk about your dissertation or your research in 1,000 words using only common words in the English language, which means you can't talk about acidification, but you can say ass water. He's mentored, I would say, dozens of undergraduates. He has collaborations that span the globe. He has a postdoc that he will step into immediately with the Smithsonian to do a continuation of his independent work that he's been doing as a PhD thesis. And in addition, he is also the one who told me what an excellent experience it had been to take grad school 640, the diversity and equity class, and that I should take it too, which I did and agreed. Also involved in writing the open letter to the department on equity issues. So I am really, really, truly proud to be Alex's academic mother and to see his development, his creative development, his independent thinking, his generosity, and his collaboration. But the whole person of Alex came to us with many of those traits already. And for that, I think he has his true mother to thank. And many of you, I think, know, but not all, that Alex's mom died on Friday. And he is going through with this in part in honor of her, because she reflected on the important history of family time together, but also looked forward without regret. And so in that spirit of reflection and looking forward, Alex.
Thanks, Jen. If you have a button, this was made by my mom, so she is here with all of us. So just keep that in mind. And what would a PhD defense be without some technical difficulties? I think people can watch this on the internet backwards. They may or may not be able to hear me. But it's good to know that they're here for me, you know? So, God, thank you all for coming. This is amazing. I'm really excited to tell you today about some work in ecological speak, thinking about how local ecosystems can modulate global change and how that influences species that are living in the system that we interact with. On a more personal note, this is about a system that we all work in, including Willoughby Bay, that produces something like 1/4 of the oysters that we consume in the United States in an aquaculture sense. So my brother, who's a chef in San Francisco, gets oysters from Washington. So this is a connected place. But in a simpler terms, I'm thinking about the system through the eyes of an oyster. And so on the right, your left, there's the native Olympia oyster, and then there's the introduced, I'm going to call it the aquaculture oyster. It comes from Japan. We brought it here for aquaculture, and its existence here depends on us. And I've been thinking about this through a couple lenses. One, through how food in the environment and food availability affects these oysters. And this is kind of carrying on work that's been going on for more than a century. But I'm bringing new tools like stabilized soaps and fatty acids to this study. And then in more recent years, people have been trying to understand the effect of pH on shellfish and oysters specifically. But today I'm going to start exploring how these things interact in the wild and hopefully fill in this gap of, well, maybe these things that the primary producers that are the base of the food web can also feedback on the environment, on the pH that the oysters experience. And I just love this picture, so I had to throw it in here, but it's important to think about the context. So people have been harvesting and consuming oysters in Puget Sound for at least 4,000 years. There's middens that date back 4,000 years that the native Coast Salish people were using this resource. And it wasn't until about 150 years ago when Europeans started colonizing and settling the area that resource became exploited, and there was something like 1 billion Olympia oysters harvested out of Willoughby Bay in a couple decades. And so when we think about the system now, it's very different. Like basically on a percentage-wise, none of the trees that were there then are here now. And so it's important to realize that context, especially when we start looking forward into the future. And a major issue that people are confronting right now, and especially in our region, is ocean acidification that happens as carbon dioxide from fossil fuel burning in the green line increases in the atmosphere. On slow timescales, it equilibrates with the surface ocean, as you can see in the red. And this is data from an open ocean site near Hawaii. So as the carbon dioxide increases in the atmosphere, it is absorbed by the ocean, and that drives the pH down. And we can measure this over the past couple decades. And really what's going on here is we have that carbon dioxide in the atmosphere, say in the pre-industrial times. And as it is absorbed, and it can go either way, it can off-gas from the ocean into the atmosphere, but it comes to equilibration. There's a balance between these different carbon species, all of which can be used by biology. So that's an important thing to know, that any of these could be changed. So plants can use carbon dioxide and bicarbonate in photosynthesis. This is a really important element for oysters because they take that up to build calcium carbonate shells. And so in the pre-industrial era, the balance was such that there was a lot of this calcium or this carbonate ion available, indicated here, and not a lot of protons or hydrogen ions. So the pH was high, and there was a lot of carbonate. As we added carbon dioxide to the atmosphere, that changed the balance of these carbon species in the ocean, and over time, that the system re-equilibrated to a point now where we have more carbon dioxide and less of that important carbonate ion. And so an analogy that's more personal is when we think of our blood pH, our bodies maintain a very narrow range of blood pH. And so within that green range, our health is high, we're kicking ass. If we go even a little bit outside of that range, our health starts we won't be feeling as good. And eventually there's a range. So even half a pH unit outside of that ideal range, we can't exist, we can't live. And so this is scary when we start thinking about the future, and we can use climate models to project out what the carbon dioxide in the ocean should be, and how that will influence pH. And we see these dramatic changes from what has been stable for many thousands of years into a rapid decline over the next less than 100 years, and a change that's approximately 0.5 pH units. And that is enough of a change to be a very big deal for aquatic ecosystems. But when we start digging into that, we see that different ecosystems have very different signatures of variability. So if we look in the open ocean over 30 days, the pH on the y-axis is very stable. And there is seasonality to it, but it's a very small range of variability. But when you get into coastal systems, like upwelling systems or estuarine systems, there's really high variability in the pH. And it could be related to an event like upwelling that brings CO2-rich water onto the shore, or salinity or tidal changes in an estuary. But we don't exactly know what is driving those patterns in the coastal ocean. And a lot of work has been done in shallow estuaries and salt marshes. And so something I was trying to do was bring some of that information into the coastal zone with the modern synthesis, which there's like a few evolution people in here, I guess. Anyway, and trying to develop a conceptual model of pH. And so in this model, we have a habitat patch and with the pH indicated by this color scale. And it's important to know that pH is constrained by the physical variables of that environment. So the salinity or the alkalinity controls the buffering capacity of that water. Temperature also influences pH. Or if that water is moving and that habitat patch is experiencing new water mass with different characteristics. And to date, we kind of think of this system as primarily driven by that atmospheric carbon dioxide exchange. And so as on decadal scales, on really long time scales, as the CO2 in the atmosphere builds up, the pH of any given habitat patch will decrease. But it's important to realize that biology can change pH too. Photosynthesis in plants like eelgrass take up carbon dioxide to build carbohydrates. And in that process, they release oxygen. So there's this trading of carbon dioxide and oxygen in photosynthesis. Similarly, we have other benthic foundation species like oysters that are taking up the carbohydrates produced by plants, using up oxygen to burn that and releasing carbon dioxide. So it's the reverse cycling. So photosynthesis is taking up carbon dioxide and releasing oxygen. Respiration is taking up oxygen and releasing carbon dioxide. And we typically think of this as happening primarily in big biomass macrophytes, the charismatic megaflora like plants, seagrass and kelp. But it's important to realize that this is also happening in phytoplankton and things that are at the base of the food web. And similarly, respiration can take place on organic matter, just detritus in the water column that's being decomposed by microbes. So as we develop this conceptual model, we need to incorporate photosynthesis that can increase the pH of a habitat patch, or respiration that can decrease. And on an instantaneous scale, those two might balance each other. But if we have biomass production, we can lock up some of that carbon dioxide for longer timescales. And so you can get increases. And as that biomass is broken down, similarly to respiration, we get decreases. And interestingly, if those two processes are decoupled in time or space, you can have large subsidies of CO2. So if you have phytoplankton production in the surface water and it sinks down and decomposes in the deep water, you might get this carbon dioxide subsidy to the deep water. And so in my dissertation, I tried to kind of bring these things together because they all have there's an ecological basis to all of these processes that might also influence oysters, more than just the direct pH effect on oysters. And so today I'm going to be talking about, in chapter 1, how and why does pH vary in the Salish Sea surface waters? And I've constrained this analysis to the intertidal range where these oysters are primarily living. In chapter 2, I'm kind of linking those large-scale patterns to the scale of aquaculture, of oyster aquaculture, and sussing out if there's a food web link. So trying to get at which biological processes might be influencing this. And then in chapter 3, bringing it home to the oysters, or otherwise known as the benthic foundation species. I got a lot of crap on Facebook for that. Okay, so starting off in chapter 1, how does pH vary in sailor sea surface waters? And this work is in a collaboration with the Department of Ecology and Padilla Bay. And Department of Ecology has been running this monthly monitoring at all of these sites, at 80 sites around Washington State since 1991. So I have a data set that, with the addition of 3 sites from Padilla Bay, is 83 sites from 1991 to 2015, where they measured pH, temperature, salinity, and dissolved oxygen. So this is a huge data set to look at seasonal and interannual changes. Importantly, the dots, if you can see them, are long-term sites where they basically have monthly data over most of that period, where the triangles were rotating stations. So they visit them for 5 years and then they switch stations. But it still gives good coverage. And so we can start looking at seasonal pH variation. The months are along the x-axis there, with pH across the y. Using those temperature, salinity, and an assumption about what the atmospheric carbon dioxide was, we can predict the pH of a water mass. And so that's what's shown here. This is the distribution of all of those 83 sites for the month of January, for example. And this is what we would expect if that water mass was in equilibrium with the atmosphere. And so just for reference, the open ocean average is a little bit higher in that top dotted line. That might be because the salinity is a little bit lower here than in the open ocean. And furthermore, the IPC projection for future ocean acidification is shown in that bottom dotted line for 2100. So this is the observed data. Each one of those points represents the average of all observations for that site in the given month. I want you guys to take a second, turn to your neighbor. If you don't know your neighbor, introduce yourself, and describe what patterns you see. Okay, let's bring it together. I don't want this to get too carried away. Can I get a volunteer? Sure. Yeah, Dad. I see. Red wine, and white wine. And white wine. Yeah, yeah. So one's a little richer, one's a little lighter. Yeah. Purple and green. Okay. What do you see relative to those predictions? If you had to bet what pH was, and I gave you these predictions, would you put your money on me? Probably not. Yeah, so one of the first things that jumps out of this is that those predictions are the predictions assuming equilibrium with the atmosphere are not correct. In fact, it's only in equilibrium with the atmosphere 6% of the observations over this entire time scale. What are some other things people noticed?
Seasonality.
Seasonality. Okay. Yeah. So in the winter months, there's not much variation between sites, and they're all lower than what would be predicted if it was in equilibrium with the atmosphere. Whereas in the summer months, a lot of the sites are higher. The pH is higher than you would expect. But then there's also more variation among sites. And we can relate this back to another variable that's important for oysters in looking at the proportion of sites with an aragonite saturation state above 1. That saturation state is just, if it's above 1, it means that it's easier to build shell. If it's below 1, they can still build shell, but it just costs more energy. So if you're a larval oyster and you drop into one of these, into Puget Sound randomly in one of these months, if you drop in February, you have about a 20% chance of being somewhere where you're going to develop normally. If you drop in May, just a couple months later, you're going to fall in a place where you're much more likely to develop normally. So there's these important, these large and important seasonality and differences among sites. So digging into that, what factors could we determine are driving these pH changes? Biological versus physical. Remember, the model that we were kind of going on initially was that the atmospheric changes are really driving these long-term changes. So we might think that that's an important thing. But to reiterate, the assumption that it's in equilibrium with the atmosphere was not a very valid assumption here. And fortunately, the Department of Ecology measures dissolved oxygen as well. And so I said earlier, when the biological process is driving the change in carbon dioxide, we should see a change in the dissolved oxygen as well. And when we plot the dissolved oxygen, we see this dotted line shows 100% saturation of the water. So that means it's in equilibrium with the atmosphere, similar to our predictions. And we see the same seasonal pattern, where in the winter, the dissolved oxygen is lower than you would expect, and in the summer, in the spring and summer, it's higher than you would expect. The points are colored based on their pH anomaly, so you see there's pretty good coherence. We actually did statistics on this too, and they confirm that dissolved oxygen is the most important variable for predicting pH change, suggesting that it's really, the pH in this system is really under biological control. And finally, I was kind of curious, like, so we have this 25 years of data, do we actually see ocean acidification in this system? And we can break it down by the regions where sampling occurred. So the coastal estuaries are Grays Harbor and Willapa Bay, Hood Canal, north of Puget Sound, and the south end of Puget Sound. In reading this plot, the dotted line is the observed open ocean kind of global average rate of change of pH. And so that's really driven by atmospheric increases. And that matches up with predictions really well. Sorry, I didn't indicate it, but this is 0 is no change. And up here would be a positive change in pH per year. And any value below that point would be a negative change in pH, or indicating ocean acidification. And this is the observed data. And so right off the bat, they're all below, sorry, the pH is decreasing faster than can be predicted or explained by atmospheric carbon dioxide changes. We also see variability among sites within the region and among regions. And so there's something else that's driving the pH down in this area. And it's related to those biological changes. So when we come back to this, we've kind of validated the use of this conceptual model for a system, because while it may not help predict the pH tomorrow, it kind of helps us understand which processes are really important and maybe where to focus our energy if we're trying to manage the pH or restore this ecosystem. And so I changed the font. It's bigger. So these biological processes are more important in this system than in some other systems. That is not to say that the physical factors are not important. They still are important, but we're just seeing the biological overwhelming them. And so the take-homes from chapter 1, photosynthesis and respiration are key drivers of the short and long-term pH change. And this is important for management to understand, because it's much easier for us to address these biological processes at a local scale than it is to address carbon dioxide in the atmosphere at a global scale. There's also the organisms living in the system are exposed to stressful pHs seasonally and even in certain locations. And again, this is important for people working in the system, either in a research, from a research perspective, or as an aquaculture or shellfish farmer. I didn't study any of this, but I have lots of thoughts. There's a lot of, it's pretty interesting to think about the implications of that. If anybody wants to talk about that later. And finally, ocean acidification in the Salish Sea is happening, and it's happening much faster than can be predicted by atmospheric carbon dioxide. So in chapter 2, I was trying to dig into more of which biological processes are affecting pH, and whether that's these kind of benthic foundation species like gillgrass and oysters that are driving large-scale pH changes, or if it's more a water column process related to phytoplankton production and decomposition. And then trying to zoom in on the scale that is applicable to people working in the shellfish aquaculture industry. And this really got started before I started grad school, in a collaboration between the Subsea Lab, who I was working with at Freddie Harbour, and Molly Roberts, who was working in Emily Carrington's lab. And she had this really interesting data that showed that when the salinity decreased in the San Juans, the pH went up. And the physical effect should be the opposite. pH should go down when the salinity goes down. So something was up there, and there's some good environmental variability there to pick apart. At the same time, I was looking at the total suspended solids, so kind of food for oysters, in the water column. And we also saw some interesting patterns where when the salinity was high, we just really didn't see very much phytoplankton in the total suspended solids. But at lower salinities, there was much more phytoplankton. And so we published a paper a couple years ago that kind of started linking these things. We didn't have simultaneous measurements, so we couldn't say for sure that the phytoplankton were driving this. But I wanted to collect more data to kind of dig into that. And so in collaboration with the Department of Natural Resources, we mapped pH in 7 important oyster aquaculture bays in Washington State. And we also, along the way, we took bottle samples to make sure that the pH we were measuring with an electronic sensor was accurate. But we also measured chlorophyll, which is an indicator of photosynthetic biomass from phytoplankton. And we also measured total suspended solids. And so this is an example of the data that we generated from Willow Bay, where we have the pH varying in the colors along a transect that we just drove around in a small boat. So it's really fun to just drive around in a small boat for hours and talk about houses on the beach. And there's a couple things that were pretty interesting from this. One, we saw a lot of spatial variability in pH, where you could have low pH right next to a region of high pH. But generally, across all of these bays that we mapped, as you get into the shallow head of the bay, especially if there's a river there, we were observing really low pHs. And just to drive that point home in a visual image that will be important later, we were seeing this in shallow kind of head of the bay, close to the river, tidal flat areas where oyster aquaculture happens. We saw lower and more variable pH. Whereas as we got closer to the ocean or the mouth of the bay, it was higher pH and more consistent. Similarly, when we looked at this ratio of phytoplankton biomass to total stuff in the water, we saw this interesting pattern where when the phytoplankton, or the chlorophyll and photosynthetic biomass was dominant, we saw high and less variable pH. But when chlorophyll was a small portion of that total suspended solids, we saw really variable pH, but that's also where we observed the low pH. And so this is further evidence to link these water column processes to pH exposure. But it's also interesting because the oysters really like that chlorophyll. They don't so much like the rest of the stuff. And so this is providing support for the idea that the food webs are linked to pH. And so in this chapter, we saw consistent patterns of pH among bays, where the low pH was kind of in the shallow end of the bay. And it's kind of adding further evidence to the connection between food webs and pH in these coastal habitats. So finally, I wanted to try and bring that back to the oysters where I initially started. I actually didn't plan on studying ocean acidification in my PhD. It's interesting how these things work. A lot can happen in 5 years. And just to remind you about how pH or how ocean acidification can affect oysters, there's this really cool work from George Waldbuster's lab, and where they measured the proportion of normal development. So they looked at larval oysters, and they measured how many of a cohort developed normally, which means just like pretty well, across a range of aragonite saturation state. And that aragonite saturation state, remember from before, is kind of a measure of how much carbonate is available in the water. And so in pre-industrial times, when there's little atmospheric carbon dioxide that's equilibrated with the ocean, we might have a value up here. Similarly, from what we saw in chapter 1, this might be in the summer season. We might have an aragonite saturation state that's high enough that most of the larvae are developing normal. But as that system equilibrates to higher carbon dioxide, it can shift to a point where very few of those oysters are developing normally. And that's kind of a stress, a physiological stress on shell building. And so when we translate that to what we might expect to see in the environment, we expect to see this positive correlation between shell length, so the growth of an oyster over time, and the pH that it experiences. So if it's grown in a low pH, it should be hard to build that shell. We should see less shell growth and elongation. Similarly, we can measure the strength of that shell, and we might assume that because it's difficult to build that shell, investments in strengthening that shell will be less in a low pH environment. And so in this study, I wasn't able to measure pH, but we were able to measure these other properties of shell, and we might assume that if pH is the main driver of these shell properties, we should see a positive correlation between shell length and shell stress. And so when we come back to this, we have this clear pH effect on oysters from lab studies, and we know that food can be important for driving physiological processes. But I'm adding this new feedback from the previous chapters, and trying to see, like, okay, so if we know that food is influencing the pH that's experienced by the oysters, food is also affecting the oysters. What's the relative balance of all of this in the field? Like, can we determine whether one of these is more important? And fortunately, Washington State has a lot of great opportunities to do this experiment in the field, because we have these estuaries with large gradients of pH. We also know from other studies that as you move along these pH or along these estuarine gradients, you have a change in food availability that goes from primarily detritus-based to primarily phytoplankton-based. And so we see these correlated or potentially correlated changes in food availability and pH. It's really difficult to measure the food that's available to an oyster over multiple months, especially even measuring what a suspension feeder is eating. And so based on previous work that I did with the subsea lab in Freddie Harbor, we found that the phytoplankton were the main sources of the fatty acids that were fueling oysters. Oh, sweet, great. It's good to know it's working. And so what we saw in that study was my access label got cut off. Sorry about that. This again is that ratio of phytoplankton to total suspended solids. So when there was very little phytoplankton relative to the rest of the detritus in the water column, there was very little fatty acids, which is an essential nutrient for all animals. But as the phytoplankton abundance increased, you saw a correlated increase in the fatty acids. So by measuring fatty acids in the oyster, we should be able to tell how much food it had available. And so I did this in 2 bays, Willapa Bay on the left and Padilla Bay on the right, and following those predicted, the pH that we observed, I put these oysters out in different environments in the bay. Just in case you thought that only food and pH were important, Bob Payne is here to remind you that predation is also an important thing in ecosystems. Snails ate all of my oysters. So, you know, it's never a simple story. So I left those oysters out for 3.5 months and then measured their shell growth and how much tissue they put on over that time span. And that's shown in here. So the sites are along the horizontal axis here. Willapa Bay 1 is the low pH area, going up to the high pH area in Willapa Bay, and then Padilla Bay is that expected low pH area of that bay too. And so in the Olympia oysters and the Pacific oysters, or the aquaculture oysters, we saw changes associated with the position along the bay. I forgot to mention that I also put them inside of eelgrass and outside of eelgrass as to kind of extend the axis of pH variability, because eelgrass as a photosynthesizer should be providing periods of time with high pH. And for both species, there was a small effective eelgrass, and it was only observed in a few of the sites. But we saw some important species differences where the Olympia oysters barely grew at all in that Willapa Bay site, whereas the the aquaculture oysters basically did fine everywhere. And similarly, when we look at how much tissue they put on, which this is an important metric for the aquaculture industry because that's what we eat. We saw similar large changes across the gradient. So the location within the bay had a big effect on the oysters, and a lesser effect of being associated with eelgrass. And again, the oysters were starving. They were actually losing mass in the river end of Willapa Bay, while the aquaculture oysters seemed to do fine anywhere that we grew them. But they did worse in that Willapa Bay location. And so we can kind of look at this from those 2 lenses of, is it a pH stress or is it a food availability stress? And these patterns are pretty consistent with both of those hypotheses. We see less shell growth and less growth overall in the low pH environments, but we also would expect the food availability to be worse there and dominated by detritus. And because we don't see a clear effect of eelgrass, and the eelgrass was only observed in kind of the higher pH areas, that's kind of inconclusive. But going back to those relationships that I talked about before, we can kind of evaluate these relationships, those effects of pH. And so remember, we assumed that shell length and shell strength would both be positively correlated to pH if pH was the main driver of that variation. And so if that was the case, you would expect to see a positive relationship between those 2 variables. We don't. So that means that something else is going on, and those 2 might be responding to separate factors. On the other hand, when we correlate tissue mass to the total fatty acid standardized per gram of oyster tissue, we do see a positive relationship for both of those species, particularly for the Olympia oysters. And there was an area, in the area where they didn't grow, their fatty acid concentration was so much lower than they were when they started that it seems like they were just using up the reserves to survive. And so it seems like there's evidence for food limitation of the Olympia oysters. So when we go back to this model of trying to evaluate if food or if pH is important in the field, we can kind of modify this. And so we know that the food is affecting the pH. We have weak evidence that there's a pH effect on oysters in the field, but we do see strong evidence for a food effect. And so to summarize that work, the food availability was a really important factor for oyster growth. And this is an important thing to consider because a lot of watershed processes influence that food availability in the watershed. So if we're thinking about managing these systems for oyster growers, we can't just think about what's in the water. It's kind of a whole watershed issue. And at least in this study, in these locations, there was weak evidence for a pH effect in the wild. And another way of thinking of this is that the pH was actually an indicator. pH could potentially be an indicator of other ecosystem processes influencing oysters. So that's kind of requires a little bit of shift in perspective. But I think at the large scale, that chapter 1 provides a lot of support for that shift in perspective, that the pH is really responding to the balance of respiration and photosynthesis in the system. And this kind of allows us to advance a new conceptual model of pH in coastal environments that really gives us power to manage things that we have more control over, things that are happening within a watershed or within a bay. And another interesting thing that comes out of this is that phytoplankton are acting at large scales. This is like the quintessential oceanography versus benthic ecology issue. Like when you stand on the shore and you look across an eelgrass bed, you're like, oh man, there's so much eelgrass bed. There's so much eelgrass here. That must be influencing pH. But when you stand in a boat offshore and you look across the ocean back to the shore, you're like, oh, there's so much phytoplankton. Phytoplankton must be. And so this is like an argument that always happens. I like to be smack dab in the middle of that. So this is a cool finding for me and where I'm going in my career. But phytoplankton are acting as ecosystem engineers across very large scales in the Salish Sea, and kind of driving a lot of that seasonal variation that we see. And that provides a link between food availability and pH exposure. And so it really argues for the need for an ecosystem perspective to ocean acidification, especially in coastal systems. In open ocean systems, they're operating differently. So that's some of the stuff that I've been doing for the last 5 years. I feel like I needed to cut the science short a little bit so I had time to thank everybody, because I know you're not here for the ecosystem modulation of ocean acidification. Okay, thanks. Thanks, Liz. Okay, so I want to start with some philosophy about luck. There's 2 types of luck in my mind. There's type 1 luck, where the odds are equally low for everybody, and you win. So like during the chapter 3 work, when I had oysters put out in Willapa Bay and Padilla Bay, I picked them up in late October. It's during the fall transition. The weather sucks. It's just like really bad weather. There was huge storms coming off the Pacific Ocean when I had to pick up the oysters. And so the tides were at 4 in the morning. Multiple days in a row, the storm broke, and we just had lovely sunsets and calm weather just for the tide. And then as soon as we got back to the house, it just started raining down. That only lasted for 2 days. At the end of that, Scotty and Julia Cobalt were with me on the objectively worst field day that I've ever had, where we almost got lost on the tide flat in Padilla Bay at 4 in the morning in like a tropical storm. And then, so that's type 1 luck, where you just get lucky. Type 2 luck is when the odds are low, but there's people working behind you, and you're benefiting from the work of others, and therefore you win. And I feel like I've had that for a long time. Like I've just been very fortunate to work with people who are looking out for me and helping me get to where I want to go. So I want to start by thanking my committee. This is Megan Detier accepting an award next to Sylvia Earle. Someone clearly has better style. That asterisk means that I've published with them. This is an academic talk after all. So while I mostly appreciate y'all being my friends, we also have to push buttons. So and so between Megan and Daniel and Paul, I feel like I have a very balanced committee that push quantitative thinking, which isn't necessarily always my favorite thing to do, but also broad thinking. And something I really admire in Megan after working with her for so many years is that she can put the pieces together. She takes these data sets and makes them make sense in a miraculous way. And Jen has been an awesome advisor. When I came to grad school, I was kind of all over the place. I wanted to study urchins catching drift algae and amphipods in Antarctica and oysters in Lowe Bay. And she was like, cool, yeah, we can make it work. But she's always reminding us of how important top-down control is. And it has interesting ways of training us on that. But it's kind of like it's the advisor's job to take a lot of shit from their grad students. All advisors do. But Jen's the only one that actually handles their shit. And so Lowe Bay. This is the composting toilet in Lowe Bay. We all joked that Lowe Bay is like a formative character building experience, and it is when you move that bucket, you become a different person. but I really like the time that I had down there. It's a unique place. But it's not just my grad committee. I mean, my mentors go back from the time in Friday Harbor to my master's degree, more time in Friday Harbor to my undergrad with working with Tessa Francis and Kenya Whitehead. And all of these people have hugely influenced me. And so I think Robin and Langdon gave me an appreciation for environmental variability and not stopping with the mean, but looking at the error bars and thinking about what's going on in there. So most ecologists see big error bars and damn. And I'm like, let me see that data. And David Duggins just taught me how to power through. That's like the most important lesson there. Kevin Britton Simmons told me, as I was like a young ecologist, and he's like, you have good intuition, trust it. And that was huge. The UW Marine Ecology folks, we're kind of a big family. There's thin partitioning between labs. And we all work together, and that has been really fun to interact with you all and think about these things. In the broader UW community, biology has an awesome office staff, and I love all the work that Eddie does. I also like that you go down there and he's singing. It just makes me happy. But I've had a lot of fun working on different biology department committees and working with GLADE. So there's a lot of people, there's a lot of grad students specifically in this department who are trying to increase that type 2 luck for a lot of people. And I really appreciate all the work that those folks are doing. My undergrad mentees, that's really been fun. I like working with people who don't know how to do science because they like challenge everything that you do, and then you realize that you're doing stuff wrong. But I had multiple opportunities to take part in Proud Mentor Day at the undergrad research symposium with Christine and Molly presenting work that they did with fatty acids and bivalves. That's really cool. The Friday Harbor community I've been part of for like 8 years, so there's so many more names that aren't on that list. And then I got to go in a submarine. And I got to look at urchins catching drift algae. So it's like, dreams do come true. And the state agencies, like none of this could have happened without the support on all levels, including financially from Department of Natural Resources. And I really appreciated working with these people, but also the way that they work. Like this was a field outing with Department of Natural Resources. Like you cannot pack more field work into a boat than they did. But working with the state agencies really helped me ground. I'm still working on it, but like ground my science in an application. And working with ecology, Julia Boss there was great. And the Padilla Bay NER provided a lot of financial support and were very helpful while I was working up there. Mental health, super important also. Scotty was a really good field hand too, and like helping me launch Flupsy. But yeah, these are like the people that have fun. Not the science is fun. But you know, and there's a lot of work that goes into raising oysters. I did none of that. I feel very lucky that I could just show up at Taylor Shellfish. They'd hand me a bag of oysters. I'd go on my way to do the science. I took a sabbatical in South Africa. Not a real sabbatical, just do the science. But like, I got to take a trip to South Africa and study something different. So it was still fatty acids, it's still bivalves, but it was a different system and kind of thinking about different influences. And that was funded by the Robert T. Payne Experimental and Field Ecology Fellowship. That was very cool. And so finally, I have to thank my family for just being incredible. My brother is the funniest person I've ever met. He's a chef in San Francisco. So if you think getting a job in academia is hard, try to be a chef in San Francisco. For many years, he's just, I can look up to him to see like how much work it takes, but how the payoff is and the motivation to keep going. And my mom came up with this joke where she was joking that she's low tech, so LWE tech. So we made these shirts because we were like redefining the term, because she's actually like way savvier than the rest of us, and like always on Facebook and playing Scrabble and stuff, and photoshopping things. And I really wanted to get this into the title of my dissertation, but I just couldn't make it happen. So, you know, there's like low class, low life, low emissions. But we redefined the term. But seriously, this is a good picture of us because with my mom's guidance, we packed way more into one frame than you can normally do. And I really appreciate that my parents always focused on education and they pushed education. And any opportunity that it furthered my education was something that they buy into. So like in 7th grade, I got a Fender Stratocaster. So I was like, hey guys, it's like important for my education. and so it's huge for me to be standing here with the opportunity to teach more people and carry on my mom's legacy as a great teacher. Because like great teachers, your students move on, but you want them to carry the lessons that you teach them. And she prepared us to do that. I also want to point out that my dad is a humble and compassionate leader, and that's something that I truly admire and try to bring to my life. And so it's, you know, it was kind of hard. There was some doubt whether I would be here today, given what has happened in the last week. There's an only boy. And you know, and like I had a lot of discussions with people who were like, wow, are you gonna do it? And there's a pretty simple answer that yes, I was gonna do it because my mom told me to. And I'm a mama's boy. And so that was the end of the discussion. And so with that, I would like to thank all of you for coming and all of the people who funded me and helped me do the research that I've done over the last few years. Thank you. It's 2 o'clock. If anybody needs to run, do it now.
I will also just remind folks that Alex can field up to 10 minutes of public questions, and then his committee will grill him. And then release him to hopefully, well, sorry. Please join us later. I'm sure an email will go out on 5th floor of Kincaid to celebrate.
Any questions? Yeah.